Thanks very much, Rob, because we've done a lot of that work with you, so you should know that. Okay, so I think it's very important to point out to, especially in Melbourne, that the first demonstration, definitive demonstration of thymus-derived T cells helping bone marrow-derived B cells to make antibodies was done 3 kilometres up the road at the Walter and Eliza Hall Institute. And this is from Journal of Experimental Medicine in 1968, Mitchell and Miller, a very historic paper. So it then took 30 years to discover T follicular helper cells, which are specialised CD4 T cells found exclusively in germinal centres within lymphoid tissue and not in blood, which makes them very hard to find in humans. And T follicular helper cells are required to provide help to B cells to produce the high affinity antibodies through class switch recombination and somatic hypermutation that Penny was just describing. So they were defined as expressing the chemokine receptor CXCR5 in lymphoid tissue, and this lineage is mainly determined by the transcription factor BCL6, and they represent a minority, a small minority of CD4 T cells in lymphoid tissue. So when a CD4 T cell is activated by antigen, it can go down a variety of lineage pathways, and when we think of antiviral CD4 responses, we tend to think of TH1, although in our experience it's quite overlooked that there are a lot of cytotoxic CD4 T cells as well. All these subsets have characteristic chemokine receptors, and they have defining transcription factors that drive their differentiation. And those transcription factors are induced by characteristic cytokines as well. So the ones we're interested in today are the T follicular helper cells that we think are driven by IL-6 and IL-21 in particular from activated cells, and they express CXCR5, which takes them to the B cell areas of lymph nodes. So in this schematic, T cells are, when an antigen enters a lymph node, the T cells are activated in the T cell area, the B cells are activated in the B cell area, and then the two sets of activated cells come together at the border. And in some cases, they will go on to form a germinal centre. So what happens in the germinal centre? The B cell takes up antigen, which it accepts from a follicular dendritic cell. The B cell processes that antigen and represents processed peptides on class 2 to the T cell receptor of the T follicular helper cell, which initiates a dialogue between the cell surfaces of the two cells. And in particular, the main one there is CD4 ligand. Any problems with CD4 ligand, such as mutations, leads to syndrome in humans called hyper-IgM syndrome, where B cells don't class switch and they don't make affinity mature antibodies. So CD4 ligand non-redundant, and also the T follicular helper cells also make interleukin 21, which is very important for B cell differentiation, but probably also feeds back to T follicular helper cell differentiation as well, and survival. Okay, so the germinal centre is a microanatomic structure within the middle of the B cell follicle, and it's polarised into a light zone and a dark zone, and in the light zone is where that cognate interaction between the T follicular helper cell and the activated B cell occurs. And so the activated B cell, after it interacts with the T follicular helper cell, moves to the dark zone where it proliferates extensively. And during that proliferation, it undergoes class switch recombination, and that's driven by an enzyme called activation-induced cytidine deaminase, which causes IgM, switching from IgM to predominantly IgG normally, or in the gut from IgM to IgA through excision of the intervening DNA between the VDJ and the heavy chain locus. At the same time as the cells are expanding, they also undergo somatic hypermutation. Again, that's an effect of AID, and that's due to random introduction of mutations in hotspots in the VDJ itself. So After the cells have proliferated, and then there's a swarm of mutated B cells come out of the dark zone back to the light zone. So all this has been watched on movies in murine lymph nodes. It's very difficult, of course, to see any of this in human lymph nodes. The mutated B cells, those with a certain affinity, accept antigen again from the follicular dendritic cells and go back to the T follicular helper cells and can initiate the whole cycle all over again. And this is the process of affinity maturation. B cells with unsuccessful B cell receptors undergo apoptosis and are eaten by macrophages. So it's not really known how many cycles B cells can go through, but at some point B cells do make the decision to exit the germinal centre, and once they do, they have to make it a fate decision between a B cell that makes antibodies, a plasma cell, or a memory B cell that's that does not secrete IgG. So, and the other important question is how long these germinal centre reactions last for. So in a mouse model, it was found that with a protein antigen in alum, the germinal centre reaction lasted between 30 and 100 days. When they used sheep red blood cells as an immunogen, the reaction lasted up to 200 days. So we're starting to get out to the length of time that Penny was talking about for requiring a lot of somatic hypermutation. So that brings us to the question of why germinal centre reaction actually stops, and is there negative regulation? So it was reported a few years ago that there are FOXP3 positive T follicular helper cells inside germinal centres, and it's possible that these are putative Tregs that may limit the germinal centre reaction. The only problem with that is that we've looked very hard for these double positive T follicular helper cells and find that there's very, very few of them, and others have reported the same. So we're not sure how much that applies. Another possibility is that, as reported at this meeting, at the last session I was at, and also at the CURE meeting, that there are CXCR5 positive CD8 T cells that enter the follicle. And the role of these cells, we think I've always presumed in HIV infection, was to kill virus-infected CD4 T cells, but it may be a more normal response to limit, to suppress any further CD4 response. And finally, activated B cells themselves may feed back negatively on the germinal centre reaction, so-called B regs, and these have been described as PD-L1 high IL-10 producing B cells that suppress T cell responses. And that's particularly been reported, the last 2 points have been particularly reported in HIV infection. So what happens to germinal centres in HIV infection and T follicular helper cells in particular? Well, T follicular helper cells, as I said, express CXCR5, but they are reportedly CCR5 negative. And therefore, back in 2008, we hypothesised that T follicular helper cells were protected from HIV infection. And our clinical experience, of course, is that HIV-infected subjects have plenty of antibodies to HIV-1. These antibodies are IgG, so they're class-switched. They're high affinity and very high titer. And as Penny pointed out, recently B cells that make neutralising anti-envelope have been reported to have large numbers of somatic hypermutations. So taken all together, there are HIV-specific T follicular helper cells in lymphoid tissue. This sort of goes against the dogma at the time that there are no HIV-specific CD4s, that they're all wiped out very early in the infection. So this is a series of serum samples run on Western blots and all put together by Philip Cunningham from our diagnostic lab, and they show the evolution of the antibody response during primary HIV infection. And you can see from weeks 2 to week 79 that there are increasing numbers of viral proteins that are being recognised, and not only that, but even up to the last few samples there's increases in titer as well, which suggests that this is a very active process. And not only that, but a couple of years, this looks old because it is old. It was 20 years ago that we did this trial of treating primary infection with AZT. This particular patient was randomised to no therapy. A couple of years later, we did an open label trial with potent antiretroviral therapy, and we found that if you treat it very, very early, then the P24 antibody response actually starts to go down. So that is an active process. And that's driven by germinal centres in early chronic HIV infection. So it's very characteristic of the histology of lymph nodes in early chronic HIV infection that there are numerous very large and irregular germinal centres in lymph nodes, that these germinal centres can disappear after antiretroviral therapy. And not only that, but if the patient interrupts treatment, the germinal centres can come back within 30 days, which is about the same time that the virus comes back. An important thing about these germinal centres is that they're absolutely covered in virus and P24. And in fact, to the point where you wonder whether, if another antigen came along, whether it would even get a look in a B-cell response. Okay, so unfortunately, we can't access lymph nodes during acute HIV infection to follow all the molecular details and cellular details. But we switched to the SIV infection of pigtail macaque model to look at lymph nodes during very early infection. And early on, we defined T follicular helper cells in macaques as PD1 high and CD127 low. I'll show you that in a minute. Because there was unable to find cross-reactive human antibodies to identify CXCR5 in the monkeys. But we were able to confirm expression of CXCR5 by PCR and also IL-21, ICOS and BCL6, and later by antibody as well. Importantly, the T follicular helper cells in the pigtail macaque lymphoid tissue were negative for CCR5, as well as the alternative co-receptors for SIV, GPR15 and CXCR6, that's Bob and Bonzo. And what we found was that the T follicular helper cells were greatly increased as a proportion of memory CD4 T cells in SIV-infected macaques. And not only that, but we purified those T follicular helper cells from lymph nodes and spleens and showed that they were infected with SIV DNA and also productively infected. So this is the flow cytometry showing you the PD1 high, CD127 low definition of T follicular helper cells. And note underneath there's a population of cells that are PD1 medium as well. But the PD1 high cells also express BCL6, which types them as TFH. So this is the proportion of CD4s that are T follicular helper cells early, that's less than 28 days, versus late, that's greater than 77 days after infection. And you can see a clear rise. The lines join longitudinal samples from individual monkeys to confirm that there's a rise. And if we just gate on memory CD4s in the lymphoid tissue, we find that something like a third of the memory cells are actually T follicular helper cells. So they really do accumulate over time. So these results were very consistent with results from 3 other groups that came out around the same time, 2012, 2013. And Yin from our group isolated DNA from those purified cells and measured by PCR SIV GAG DNA and found that the PD1 high cells were indeed infected, and infected as much as, or if not more than, other subsets in the same lymph nodes. And not only that, but Yin managed to amplify envelope from the same DNA and cloned and sequenced, and we could not find any characteristic signature of the PD1 high cells. All the sequences were mixed between all the different subsets, suggesting that there was no one strain of virus that was getting into the TFH. And then we collaborated with Professor Brigitte Autran's group in Paris. They had cryopreserved splenocytes from chronic HIV positive patients. And in Paris, they sorted them into the various subsets, particularly the germinal centre T follicular helper cells. And again, they sent us the cell pellets, and Yin did the HIV DNA assays and found that the germinal centre T follicular helper cells were more infected than other subsets. And not only that, but they again looking at envelope sequences, by geno to pheno, they look like they were CCR5 using, which we'd already found these cells don't have CCR5. And also these results are consistent with two other groups that looked at human TFHs and published in 2012, 2013 as well. So what we think is happening is that the antigen-activated CD4s first go to a PD1 medium CCR5 positive phenotype before they branch out into the other subsets. And if SIV is around, then it's highly feasible that these cells are infected. So what Yin has done, and it's on her poster, I don't know the poster number, but I think it's tomorrow, that SIV from PD1 medium cells were highly purified from SIV-infected lymph nodes and then incubated with anti-CD328 and IL-21 for 3 days to push them down a T follicular cell pathway. And they induced high levels of markers of T follicular helper cells, but they also took with them SIV DNA. So that's one plausible explanation of how T follicular helper cells are so heavily infected. But that brings us to the question of how then do we manipulate any of these processes to improve vaccines? So the obvious first target is interleukin-21. That's already been extensively trialled in humans because it has another effect, and that is to boost CD8 and NK cell activity in cancer patients. So there's one published report and there's another report from this meeting that IL-21 can be given to SIV-infected macaques and it boosts CD8 responses. And in the first report, published report, it was shown that the anti-SIV antibody titers were boosted by the IL-21 treatment. Another very hot topic in cancer therapy is PD-1 blockade. So there are plenty of reagents being worked on for that. And the rationale here would be that PD-1 blockade may help the B cells, the T follicular helper cells, to signal to the B cells. In HIV positive lymph nodes, it's been reported that the B cells overexpress PD-L1 and that this inhibits TFH function. So PD-1 blockade may actually help TFH responses. The only problem is in monkeys, in mouse models, it's been suggested that the PD-1 signalling back to the TFH may drive IL-21 or may increase IL-21 production. So it's not really clear how that works, but definitely empirically in at least 2 reports, PD-1 blockade led to increase of SIV antibody titers, anti-SIV antibody titers by about 2-fold. The last possibility is, as we heard in the first talk, interleukin-6 is increased in some inflammatory situations, but most therapeutics for IL-6 in humans are designed really to block this IL-6 effect on inflammation rather than enhancing it. The last possibility is to look at precursors of T follicular helper cells in blood. So what happens when a germinal centre reaction finishes? Do the T follicular helper cells turn into memory cells? So if you look in blood, about 10% of circulating memory CD4 T cells have CXCR5 on their surface. These are a very heterogeneous population of subpopulation of CD4 T cells though, and not all of them help B cells. So it's not really clear at this stage the relationship of these circulating CXCR5 positive cells to T follicular helper cells. But nevertheless, there may be an opportunity to take cells from blood, maybe to boost a secondary, boost them and generate a secondary B cell response. We don't really know at this stage. So in conclusion, if the aim of a vaccine is to generate potent neutralising antibodies, and I guess I've always been thinking along the lines of those long CDR3s that are generated from a shorter CDR3 in the germline that Penny was talking about. But I guess any affinity maturation of starting with a long CDR3 as well, like the second antibody that Penny was talking about. We really need to better understand T follicular helper cell differentiation and opportunities to modulate it. One thing though that is very unclear to me is identifying CD4 epitopes in the HIV envelope to include in a vaccine. I only know of one report of a really clear HIV envelope epitope. I presume there must be others, but it's very difficult to measure HIV-specific CD4s at any point, and usually we look at gag responses rather than ENV responses because we simply can't find them. So the other consideration is if we make more TFHs, we've got to be careful that they don't become more substrate for the virus. And on the other side, as Penny pointed out, we've got to optimise the vaccine for the envelope-specific B cells, but it has to be coordinated with the vaccine for the T follicular helper cells. And then, of course, as Penny and before her, Tony Fauci pointed out, we've got to direct shepherd B cells through multiple rounds of somatic hypermutation towards an effective, broadly neutralising antibody. So I guess we can say that there's cautious optimism now because we think we know where we need to get to, and so we just have to do it. And I'd like to acknowledge all the people, particularly Yin and Chris Weatherall before Yin, that worked on our T follicular helper cell projects and our collaboration with Rob DeRosa, Stephen Kent's lab in Paris, and also more recently with Michael Roche and Paul Goria here in the Burnett Institute on envelope sequencing and funding. Thanks.